Hi everyone, welcome to the channel. In this video we will be discussing about targeting tau in Alzheimer's disease. So these are the topics that we will be covering in this video. So first we will be discussing about healthy brain. So healthy brain, it contains about 86 billion neurons. Okay, now what is the structure of neuron? Neuron is So this is the axon terminal, or we can say axon end. This is the nucleus and this is axon. Now what happens here is this nucleus, it produces neurotransmitters. These neurotransmitters, these are to be transported to this end to transfer the signal to the next neuron. So these neurotransmitters, they travel through here via microtubules. Okay. Now these neurotransmitters are transmitted along with mitochondria through this microtubules to the axon end. Now what they do here is, I'll draw this particular structure here. For example, these are the two ends of the axons, Snare Complex. Now what is a Snare Complex? When the neuron transmitter is transmitted here, then it needs to be transferred to the another end of the neuron. So here, after reaching here, this neurotransmitter, it forms this bud-like shape, and then it is released from here and it is transmitted to another end. For this process is highly energy requiring. So this energy is provided by mitochondria. So due to this reason, these mitochondria along with neurotransmitters, these are transmitted to this particular end. Alright, now this here Snare Complex is present. This is about the Snare Complex. Next we will be discussing about microtubule structure. Now microtubules, these are like microscopic rods like structure, or we can say microscopic tubes which are stacked together. Now these tubes, they are supported by tau protein. These tau proteins, they are attached to this portion. Tau protein, they provide support to the microtubules. Okay. So now we'll be drawing these are microtubules. Now the structure of tau protein. This is repeat domain. This is the N terminal and this is the C terminal. So these are the tau proteins which are present on microtubules. They provide support to these microtubules, and these microtubules, these are used for neurotransmitter transferring, and mitochondria are also transferred via the same route. Now next we'll be studying about the abnormal tau aggregation, which this was all in the healthy, this all was in a healthy brain. Now we'll be discussing what happens in case of abnormal, or we can say disease brain. Abnormal aggregation. Okay. Now due to aging, waste increases in neurons and clearance of the waste, it decreases. This results into increase in lipofusions in the neurons. Now tau protein, they bind to these lipofusions and they form oligomers. Now when tau protein they bind to lipofusions, what exactly happens here is, earlier the structure of tau was this. This was repeat domain, this was N-terminal and this was C-terminal. Now after this binding of tau protein with the lipofusions, the structure, it changes to hairpin-like structure. So this is now the repeat domain and this is the C-terminal and this is the N-terminal. Now it forms a curved structure. Now due to this, the other tau proteins, they stack over this and they form oligomers. Okay. Now these oligomers, they form PHF, which is they further form tangles. Now these tangles, they are aggregated in the neuron. These tangles, these are aggregated in the neuron, and due to this reason, three things take place. First one is MTs, these are disrupted. Second one is MTs microtubules.
Paired
Second
helical
one is
filament.
Mitochondria are dysfunctioned. Then is Snare complex. Is disrupted. Now when snare complex is disrupted, as I told, this will not allow this neurotransmitter to release and to transport to another neuron. So due to this, signaling will not take place. So this was all for the video.